This chapter is really about one of biology's biggest design principles, the balance between division, reduction, and continuity. Mitosis keeps the chromosome number stable. Meiosis cuts it down. Together they make growth, reproduction, and inheritance possible. The title already points to the two pillars, cell cycle and cell division. Mitosis is the equational division blueprint. The trigger is simple. Cell growth disturbs the nucleocytoplasmic ratio and division restores balance. That is why mitosis exists in the first place. It supports growth, repair, and genetic fidelity. The slide also gives the important exception. While mitosis is usually restricted to diploid cells, some lower plants and social insects can even perform mitosis in haploid cells. Meiosis is the reduction division. Its logic is opposite to mitosis. If only mitosis existed, chromosome number would double every generation and the species would become unstable. Meiosis solves that problem by producing haploid gametes, and fertilization restores the diploid state. That cycle is what keeps a species stable across generations. Meiosis follows 4 golden rules. First, there are 2 cycles, Meiosis I and Meiosis II. Second, DNA replicates only once before Meiosis I begins. Third, homologous chromosomes pair and non-sister chromatids exchange material. Fourth, the final output is 4 haploid cells. The big trap is right there too. The cell divides twice, but DNA replicates only once. Prophase 1 is not a short stage. It is a marathon, not a sprint. It is subdivided into leptotene, zygotene, pachytene, diplotin, and diakinesis. The mnemonic
लता जरा पानी दे दे.
is a good memory anchor. Leptotene, zygotene, pachytene,
डिप्लोटीन.
diakinesis. This is the most complex phase of meiosis because chromosomal behavior changes step by step. Leptotene and zygotene are the opening acts of prophase 1. In leptotene, chromosomes begin to condense and become gradually visible under the microscope. In zygotene, homologous chromosomes begin pairing, a process called synapsis. The synaptonemal complex helps this pairing happen, and the newly formed paired structure is called a bivalent or tetrad. Pachytene is where crossing over happens. The tetrads are now clearly visible as 4 individual chromatids, and the exchange of genetic material takes place between non-sister chromatids of homologous chromosomes. The physical sites of crossing over are called recombination nodules, and the enzyme that drives the process is recombinase. The high yield point is crucial. The exchange is always between non-sister chromatids, never sister chromatids. Diplotane and diakinesis complete. Prophase 1. In diplotene, the synaptonemal complex dissolves. Homologs separate except at crossover points, and the X-shaped chiasmata become visible. In some vertebrate oocytes, this stage can last for months or even years. In diakinesis, chiasmata terminalize. Chromosomes condense maximally, the nucleolus and nuclear envelope break down, and the meiotic spindle assembles. That means the cell is fully prepared for the first meiotic division. Meiosis I is the division that splits homologous pairs, not centromeres. In metaphase I, bivalents line up on the equatorial plate and microtubules attach to homologous kinetochores. In anaphase I, homologous chromosomes separate to opposite poles. In telophase I, the nuclear membrane and nucleolus reappear and cytokinesis forms a dyad of cells. The red warning on the slide matters. The centromere does not split in anaphase 1. If you split the X there, you lose the mark. Between meiosis I and meiosis II lies interkinesis, a brief transition stage. The chromosomes may show a little dispersion, but this is not a full interphase. Most importantly, there is no DNA replication during interkinesis. That is the trap. DNA was already replicated once before meiosis I, and it will not replicate again in the same cycle. Meiosis II is much more like mitosis. It starts immediately after cytokinesis, usually before chromosomes have fully elongated. In prophase II, the nuclear membrane disappears and the chromosomes compact again. In metaphase II, chromosomes align at the equator and microtubules from opposite poles attach precisely to sister chromatids. It is the mitosis lookalike phase of meiosis. Anaphase II is the actual split. The centromeres of each chromosome split simultaneously. Sister chromatids separate, and shortening microtubules pull them toward opposite poles. In telophase II, the chromosomes are enclosed by a nuclear envelope again. Cytokinesis follows, and the process ends with 4 strictly haploid daughter cells. That is the completion of the reduction journey. The diagnostic matrix comparing meiosis I and meiosis II is one of the cleanest revision tools in the chapter. Prophase I is complex, with 5 distinct stages, while prophase II is simple and similar to mitosis. In metaphase I, bivalent pairs align at the equator, while in metaphase II, individual chromosomes align. In anaphase I, homologous chromosomes separate, while in anaphase II, sister chromatids separate. And the centromere behaves differently too. It stays intact in meiosis I, but splits in meiosis II. The significance of meiosis is the deeper reason this chapter matters. Meiosis preserves the species' chromosome number across generations because halving in gametes balances restoration during fertilization. It also creates genetic variation, which is the raw material for evolution. So meiosis is not just a cell division process. It is the engine of heredity, stability, and evolutionary change all at once.